Okay, well, it's a real honor for me to be here at all, and especially for me to represent my class here at these research briefings. So I study interactions between the gut microbiome and the immune system. And so what I'd like to do in my talk today is to first of all talk a little bit about how the microbiome field evolved. As I'll show you, it really didn't exist in the early 1990s or mid-1990s when I joined the field. And then I'll talk a little bit about some of the immunological questions that have surrounded the microbiome early on and how we've addressed some of those questions. So just by way of introducing the topic, so a staggering number of bacteria inhabit the human body. So for roughly every one human cell, you have anywhere between 3 to 100 bacteria. Depending on how you do the calculation. So most of these bacteria live in your intestines, but there are actually bacteria that colonize virtually every body surface. So needless to say, given their numbers, these bacteria have profound effects on human health. So despite the fact that there are so many bacteria and they obviously are likely to profoundly impact human health, there was very little interest in this question in the early 1990s, which was when I was graduating with my PhD and thinking about topics to explore in a postdoctoral fellowship. So this is a graph which I generated a couple of days ago, where basically what I did was a PubMed search by year on 3 search terms: commensal, microbiota, and microbiome. And what you can see, I think that what this illustrates is how rapidly this field has grown and where we started in the not too distant past. So you can see that we've had a rapid spike in publications in this area. Now this is when, and I should mention that Margaret McFall-Nye gave the class 4 a research briefing last year. She had a similar graph in her presentation looking at the appearance of the word symbiosis in the literature. So I think it illustrates the same point. So this is when I started my postdoc in Jeff Gordon's lab at Washington University, and he was really, and he's a member of the Academy as well, and he was really beginning to think about this question of how commensal bacteria might influence host biology. But at the time, as I said, there was very little interest in this field, so it seemed like career suicide at the time. All the more so when I joined the faculty of UT Southwestern in 2003, so I'm grateful to my institution for actually hiring me. So clearly things have accelerated. There's a great interest in this field, so much so that there's now actually a website put together by Jonathan Eisen at the University of California, Davis, where he gives out an award every so often for overselling the microbiome. So this is usually calling to task some popular news outlet that has oversold some impact of the microbiome, such as it impacts your dreams or something like that. But at the time, when I started in this field early on, we were thinking about very basic questions. There was a lot of low-hanging fruit, really interesting basic questions to be answered. And so I feel lucky that I was there at the beginning. One thing I want to mention before I talk about the sort of questions that we tackled early on is actually the idea that resident microorganisms might be essential for health actually originated a long time ago with Louis Pasteur, with the great Louis Pasteur. So he actually sort of proposed a hypothesis in a monograph that he wrote in 1885, where he said that if I would have the time, I would undertake such an experiment with the preconceived idea that under these circumstances, life would become impossible. And the experiment he was referring to was deriving animals into germ-free conditions. So this was one of the rare instances where Louis Pasteur was actually proven wrong. But it wasn't until about 50 to 60 years later that he was proven wrong. Basically, in the 1940s, people were in fact able to derive animals, including rodents, into completely germ-free, microbiologically sterile conditions. So Pasteur was wrong about symbionts and commensals being absolutely essential for life, but he was right about the idea that they impact host health. And subsequently, germ-free animals have become essential tools in microbiota research because they serve as the foundation for experimental systems that allow us to assess the impact of these microorganisms on areas of physiology like immunity, development, and metabolism. And I have shown you two pictures here. Here's germ-free research in the 1940s. This is a photograph of one of our isolators in my germ-free facility at UT Southwestern taken about 3 weeks ago. And you can see not much has changed. So this is still a fairly low-tech operation. But one thing one thing you might be wondering is why do we put up with all these microorganisms? Why do we have trillions, harbor trillions of microorganisms and put all the energy into maintaining them without having them cause disease? And so one thing people noticed when they derived animals into germ-free conditions is that these animals actually had to eat more to maintain their body weight as compared to their normally colonized counterparts. And this is because it turns out that the emerging view is that the primary function of our intestinal bacteria is metabolic. So they function in large part to help us degrade polysaccharides that we take in as part of our diet. So if you're eating plants as part of your diet, these have complex polysaccharides. We're not very good at degrading them, but our microbiota, our microbiome is. And so you can see this if you look at the genomes of the organisms that normally colonize the human intestines. So if you count up all of the carbohydrate degrading enzymes encoded in the human genome, there are about 95. But if you look at a sort of a prototypical gut commensal, Bacteroides theta iota omicron is what I'm showing here, and this is one of my favorite organisms. I've been working on it for nearly 20 years now and recently learned the proper pronunciation, as it turns out. But so B theta, as we call it, has about 251 carbohydrate degrading enzymes encoded in its genome. And so if you consider the fact that the size of the human genome is about 3 orders of magnitude greater than that of B theta, it's really dedicating a large proportion of its genome to the process of carbohydrate degradation. So we're getting a lot of metabolic firepower from our gut microbiome. And so essentially what we've done over the course of millions of years of evolution, of evolutionary history, is to acquire a readily adaptable and rapidly evolving external genome that harbors a diverse array of carbohydrate digesting enzymes. And so what this does is it endows us with the ability to hydrolyze otherwise indigestible polysaccharides, basically liberate sugars that we can then uptake, and maximizes extraction of calories from the diet. The other thing that it does, though, is it allows flexible adaptations to dietary changes. So if you start out eating bok choy and then later you start eating bananas, your microbiome is part of what allows you to flexibly adapt to this dietary change and efficiently extract calories from these different food sources. So clearly there's a symbiotic relationship between us and our microbiome based on these metabolic capabilities. But these microorganisms, despite the fact that they have a purpose, are still capable of causing disease. If they're in the wrong place at the wrong time, you can get terrible disease such as inflammation, sepsis, and things that can actually cause death if left unchecked. So it sort of raises the question, if you've got trillions of bacteria in your gut, why don't your bacteria more often make you sick? Why are everybody in this room is basically healthy, and why is that? Because we all have a large bacterial population that could run amok. So that's basically the question that has driven the research in my lab over the last 14 years. So the way we've approached this question, so it's an immunological question at its core. So the way we've approached this question is to focus on a specific set of cells that line the intestinal, the inner surface of the intestine. So you can think of the intestine as an inner tube, and its inner surface is lined with a single layer of epithelial cells. So if you split this open and lay it out and look at it under a microscope, you can see that the intestinal surface is covered with a single layer of epithelial cells. So this is basically all that's standing between you and roughly 100 trillion bacteria. So our thinking was that these cells are probably very important in understanding how we maintain symbiosis with our gut microbiome. And so that's why we decided to focus on these cells. And so this was, when I started my lab in 2003, of course, new functional genomics tools had appeared on the scene. We had DNA microarrays at the time. And so we deployed these tools in conjunction with germ-free mice to explore how gene expression in epithelial cells is regulated by the microbiome, thinking that if we could identify microbiome-inducible genes, there might be genes encoding proteins that are important for defense of the epithelial surface and preventing bacteria from entering deeper tissues. And so one of the hits from our initial functional genomics screen that proved to be fruitful in this regard and really gave us insight into how this whole system works was a gene encoding a protein called Reg3 gamma. So this stands for regenerating islet-derived protein gamma. This is based on the fact that it was originally discovered in the pancreas, but actually the site of its highest expression is in the intestine. And it turns out, so this is the crystal structure of Reg3 gamma. This was solved both by us and another group. It's a small protein, 15 kilodaltons, and it's basically secreted by the surface of the intestine into the lumen of the intestine. And so you would expect it to come into contact with the bacteria that live there. And in fact, what we discovered through a series of detailed biochemical experiments is that this is a protein antibiotic. So what it does when it encounters bacteria is it tears holes in their outer surface. So this is an electron micrograph of Listeria monocytogenes, which is, it's actually a pathogen. You sometimes can get, it's a foodborne pathogen that you might get in something like cheese or potato salad. So if you add Reg3 gamma, purified Reg3 gamma protein to these organisms, you can see that they don't fare so well. They've got holes in their outer surface. And you can see their cytoplasmic contents are leaking out. So that led us to the question of how this gut antibiotic seeks and destroys bacteria. So we spent several years unraveling the biochemical mechanism of this. And I'll just summarize sort of what we found. Basically, we found that there are two steps. The first step is that the protein, which is secreted from the epithelium into the intestine, has to identify its bacterial targets. And it does this by binding to sugars that are located on the outer surface of the bacteria. So Reg3 gamma sees these sugars, so it's a sugar-seeking missile. The way it actually kills the bacteria, though, requires a different activity, and this involves forming pores in the bacterial membrane, which is actually protected by the sugars that I just talked about. So it kind of penetrates through that sugar layer, comes into contact with the bacterial membrane, and assembles into a pore. And this is a electron microscopy of the human form of Reg3 gamma, which is called Reg3 alpha, in association with artificial lipids. And we've determined a fairly high resolution structure of this pore complex by cryo-EM, which is shown here. And the time went very fast, so I'm going to speed up and tell you what I think is the most interesting part of my story. So we know a lot about how this protein kills bacteria at a biochemical level. But the bigger question was what's actually going on in the mouse or in a human. So how is it actually defending the epithelial surface? So to understand this question, I'm going to revisit the surface of the intestine, which I showed you before, and explain that not only is there an epithelial layer between you and your microbiome, but there's another physical barrier, which is a layer about a 150 micron thick layer of mucus that overlies the whole surface of the intestine. It sort of acts like flypaper in terms of preventing bacteria from accessing the surface of your intestine. They sort of get stuck in this outer layer. And so the punchline of what I'm going to tell you basically is that Reg3 gamma is secreted from the surface of the intestine into this mucus layer and sort of acts like a little landmine and helps keep bacteria from accessing the epithelial surface. So our insights into how Reg3 gamma does this were actually preceded into some basic insights about how bacteria are positioned relative to the surface of the intestine. And so this involved a technique called fluorescence in situ hybridization, where we label bacteria with a fluorescent probe that is designed to hybridize to a gene. It's not too important what the gene is, but it's a gene that's specific to bacteria. So here what we've done is taken a slice through a mouse intestine and then added this probe, and what it does is it lights up the bacteria. Here's the small intestinal tissue. It's been counterstained with a DAPI counterstain, so the nuclei are lighting up, and it basically gives you the outline of the tissue. So what we immediately noticed was that the bacteria labeled in green are sort of kept at arm's length relative to the surface of the intestine. And so this really wasn't fully understood before. It was thought that bacteria were actually in direct contact with the epithelial surface. But there's this clear zone, and just to give it a name so that we can talk about it, we called it the demilitarized zone. And this term is actually entered into the literature now somehow. But it's a very good description of what we see. And so coming back to Reg3 gamma and its role in protecting the epithelial surface, we asked a series of questions regarding how the immune system might help maintain this DMZ, and that led us back to Reg3 gamma. So again, it's an antimicrobial protein that's secreted from the surface of the gut. And what we noticed is that when we knocked it out in mice, so these are mice that are genetically deficient in Reg3 gamma, compared to wild-type mice. And you can see that the wild-type mice maintain a DMZ at the surface of their gut, but the Reg3 gamma knockout mice don't. So that told us that Reg3 gamma is important for limiting contact of bacteria with the epithelial surface. And so this is a very important concept in terms of understanding how we maintain symbiosis with the microbiome. By limiting access of 100 trillion bacteria to our tissues, you can see how you would limit their pathogenic effects. And so essentially what we've discovered, and I didn't discuss all of the data that back up this model, is that there's a negative regulatory feedback loop at the surface of the gut where bacteria are sensed through receptors that see bacteria-specific molecules. This turns on Reg3 gamma, this protein antibiotic, which is then secreted into this inner mucus layer, sort of like a little landmine, helping to prevent bacteria from binding to and entering our tissues. So this is a very simple story illustrating how we maintain symbiosis with a very complex and sometimes unpredictable bacterial population. My last slide is not really to make a scientific point, but rather just to say things are a lot more complicated than this. So the microbiome is incredibly complicated. The immune system is incredibly complicated. A lot of people are working on this now, and now we have diagrams like this in review articles are almost impossible to understand. But I think that the application of of many new experimental tools to old experimental tools such as germ-free mice should lead to many exciting new discoveries in this area, and I'm happy to be a part of it. So I'll stop there and thank the many wonderful trainees that have passed through my lab in the last 13 to 14 years. They've all been, I'm so deeply appreciative of their energy and their creativity and their persistence. And none of us would be where we are today without our trainees, that's for sure. I'm also very lucky to be at an institution where I had an incredibly supportive matrix to grow my research program over the years. And I won't name them, but I've had many collaborators at UT Southwestern who helped push my science forward and helped me raise my game, which was very important as well. So I'll stop there and thank you very much.
I think we've used up our 5 minutes. Jerry, one question and that's it. It's the last question you're going to have today, too.
You showed us how it can kill bad
Actually,
bacteria.
there's microphones there, Jerry. Go use the
Well, I
microphone.
think that people can hear me.
I can hear you. I'll repeat your question.
You showed us in detail how it can kill bad bacteria, but you didn't tell us how it decides which are the bad ones and which are the good ones.
This is a really great point. So this is, so you would think that these innate immune effectors would distinguish between good and bad bacteria, but it's not on the basis of good and bad. So there's no way to tell intent of a bacterium. What you can tell, though, is where it is. If it's too close, it's bad. It doesn't matter if it's a commensal or a pathogen. If it's juxtaposed to the epithelial surface, you want to get rid of it. So these molecules don't distinguish between good and bad.
Okay. Thank you.
Thank you.
Good, Jerry. You asked the one question I think most of us in the audience were curious about. Our second